Alright, hello. This week we are going to be taking a look at the circulatory system and the lymphoid system. So let's talk about the circulatory system first. We're going to look at blood vessels of different sizes. So let's start with capillaries. In this tissue section you can see several capillaries, these structures here. Capillaries are only about as wide as the red blood cells in their lumen. So here you can see a red blood cell or erythrocyte, and here's the capillary surrounding it. Here you can see a couple over here that are a little bit tighter around the red blood cell. So the red blood cell takes up almost the entire space of the lumen. Capillaries are essentially composed of endothelial cells, and you can see the nuclei of those endothelial cells are darker, more basophilic structures, and there are also associated pericytes which have their nuclei a little bit more to the side, such as the cell right here. This is capillary shown in longitudinal section in cardiac muscle, and so in this section you can see again, the lumen of the capillary is only about as wide as the red blood cells, and so they're forced to travel single file through the capillary. Here's a slightly larger blood vessel. This is probably a post-capillary venule, and you can see maybe two at a time can pass through the capillary in this particular case. In this section we have a couple slightly larger blood vessels. Here on the right you have an arterial, and on the left you have a venule. And you can notice the difference between these two structures. The arterial has a more regular shaped wall to it. It's more regular and circular shape. The venule has a wider lumen, a thinner wall, and because the wall is thinner, it tends to lose its shape and becomes kind of your regular shape when it's sectioned. The arterial here has several layers of smooth muscle in one of the layers, which is known as the tunica media of this blood vessel, and so this dark red tissue here is the smooth muscle. Arteries have and arterioles have more muscular walls than associated venules and veins, and so that's why they help to maintain their shape a lot better. In this section you can see a small artery here. Just a couple probably arterioles over here, and then this would be a small vein. Notice the size of the red blood cells look quite a bit smaller. Again, the small artery here has a more regular shape to it, thicker muscular wall. In this case there's at least 5 layers of smooth muscle, which is why we would classify this particular blood vessel as a small artery. Here is the small vein that has a couple layers of smooth muscle on its wall, but again the tunica media is not quite as well formed. Here again is another section in the submucosa, the jejunum. Here's a small artery, here's a small vein, here's another pair of this might be an arterial and another small vein. So one nice thing is that arteries and veins, or arterioles and venues, often tend to travel in pairs. So if you can identify one of the pair, you can often see the other in the pair very close to it in that same section. In this section we have a medium artery on the left and a medium vein on the right, again in pairs. This particular section has been stained with an elastic stain that actually lets you see regions of elastic tissue within the wall structure of the blood vessels. Arteries have more elasticity because they're under greater pressure. Typically you need to stretch more than veins, which are lower pressure and don't tend to have as much elastic tissue. Some veins do have more elastic tissue. So here again on the right we have a medium artery, and on the left there's a medium vein here. And the way we use to distinguish between small, medium, and large veins and arteries and arterioles and venules is really by approximating the number of layers of smooth muscle within the tunica media of these various structures. So reference your laboratory manual to get a guess at that. So medium arteries vary pretty considerably in the width of the muscular layer of the wall, and so based on the table in your lab manual, they have between about 10 to 40 layers of smooth muscle in the tunica media here. The tunica media of the small vein is much smaller in comparison. The outer layer, the Tunica Adventitia, is very well developed in veins, and you can see some elastic fibers in the Tunica Adventitia, both of this median vein and the median artery here. Here's a higher magnification view of a medium artery, and with this particular section you can really see the 3 layers of the wall of blood vessels very nicely. So the innermost layer is known as the Tunica Intima, which is this layer right here, and it actually consists of several parts. You have the endothelium. Endothelium is always a single layer thick because it's always composed of a simple squamous endothelium, and so the superficial most layer of the Tunica Intima is adjacent to the lumen. Now at this particular magnification, the endothelium is really hard to distinguish from the other layers of the Tunica Intima. Underneath the endothelium you have some subendothelial connective tissue, and just deep to that you have what is known as the internal elastic lamina, which is a sheet of elastic connective tissue that's found in the Tunica Intima. It's the 3rd layer. So in this particular slide, the internal elastic lamina appears dark, kind of a blue-black color, and it because it's not straight, has kind of a wavy appearance to it. So typically internal elastic lamina is only found in the Tunica Intima of arteries and not in veins. So deep to the internal elastic lamina marks the border of the tunica media, and the tunica media again has several parts to it. Mostly it consists of multiple layers of smooth muscle, between 10 to 40 layers of smooth muscle, which you're seeing here. There can also be some elastic fibers throughout the tunica media, but most of those elastic fibers are again concentrated in another lamellar structure, this time called the external elastic lamina, which is similar in appearance to the internal elastic lamina but much thicker. And so the internal elastic lamina is actually part of the tunica media of arteries, but again is not typically present in veins. Some of the larger veins can actually have more elastic fibers in their walls and may have elastic laminae, but most veins do not. And then finally, outside of the external elastic lamina, which marks the deepest border of the tunica media, you have the Tunica Adventitia, also called Tunica externa, and these structures are both very well formed in both veins and arteries. In veins, there's really not as well of a defined tunica media, but there's a much larger Tunica Adventitia. Here's a section of a wall of a large artery, and we're only going to look at one large artery in lab, which is the aorta, which is the largest artery in the body. And you can see the large number of layers of smooth muscle here. This is a relatively low magnification view, and this is all the tunica media. Here's the Tunica Adventitia or externa, which is paler and more fibrous in appearance, and then the Tunica Intima is well developed as well, but you need to look at it at a closer magnification to be able to distinguish it from the other layers. So this particular blood vessel is so large that the entire section won't fit on the slide. So you only have part of the wall here of the aorta, which has basically been flattened out. Here's a higher magnification view of the interface between the tunica media here and the tunica adventitia or tunica externa. And on this particular slide, you can see that there are actually blood vessels of various sizes, both veins and arteries, or arterioles and venules, within the tunica adventitia. These structures are called vasovasora, which means blood vessels within the blood vessel. And you find these in the outer parts, the outer walls of large arteries and large veins, mainly in the tunica adventitia, but you can also find them sometimes in the outer portions of the tunica media. You find these structures in large arteries and veins because the outer portions of the walls are so large that they can no longer rely on getting their nutrients from diffusion from the blood that's being carried way up here in the lumen. And so these outer layers of the wall have their own blood supply because the tissue is so thick. Here's the example of a large vein that we're going to look at in lab. This comes from the, I believe, the inferior vena cava, and the tunica media and the vena cava, while much thicker than in medium and other smaller veins, is not nearly as large or well developed as in large arteries such as the aorta. So it's not nearly as large of a structure. Most of the width of the wall of this particular type of blood vessel is all tunica adventitia, which oftentimes contains smooth muscle with scattered throughout the connective tissue here, which helps to propel blood along the vein. The tunica intima in large veins is not particularly well defined and oftentimes can be hard to distinguish it from the underlying tunica media. So be able to identify large, medium, and small arteries and veins, as well as arterioles and venules and capillaries. And again, the way to do that is to really try to count the number of layers of smooth muscle in the tunica media using the guidelines that are set out in the lab manual. And so, one way you can do that is kind of, if you can see nuclei in the layers, you can kind of count the nuclei to get a rough estimate of the number of layers. So moving along, this is a section of the inner part of the heart. We've looked at this before early on in lab. This, as you may recall, this is a layer of the heart is called the myocardium, and it contains the contractile cardiomyocytes, which stain a deep reddish color. This would be a chamber of the heart where blood is stored, and so this layer right here is referred to as the endocardium. The endocardium has multiple components. The superficialmost layer of the endocardium is composed of endothelium, again, which is composed of simple squamous epithelium, the same type that lines all of the blood vessels within the body. Deep to the endothelium, you have the subendothelial layer here, which contains connective tissue primarily. Many of these fibers are type 1 collagen fibers, which in this particular section are stained a bluish color. Deep to the subendothelium, you have this layer here, which is often referred to as a subendocardium. Although it's called subendocardium, and it may make you think that it's beneath the endocardium, it's actually considered to be part of the endocardium, but it is the deepest portion of the endocardium. And this layer is composed primarily of Purkinje muscle fibers as well as connective tissue. Here's a closer look at the endocardium. Again, the superficialmost layer is composed of an endothelium. Here you have the subendothelium with lots of blue staining collagen fibers, and then here's the subendocardium that contains the Purkinje fibers. Purkinje muscle fibers are non-contractile, so you can often see the nuclei pretty well, but they appear to be very pale staining inside. That's because they don't have contractile myofilaments on the inside of their cytoplasm, so their cytoplasm appears to be pale or kind of empty. These Purkinje fibers play a role in fast conduction of electrical impulses through the myocardium, and so they're specialized non-contractile cells. That's why they appear different. These Purkinje fibers you find close to the inner surface of the heart, so if you want to identify the endocardium, you should look for these Purkinje fibers. Recall that the heart actually has two surfaces. You have the inner surface lining the chambers of the heart, as you can see here. That's where you have the endocardium. The other layer is called the epicardium, and it looks like this. So this is the outside of the heart, which normally would be covered in a pericardial sac. Here is the myocardium towards the outside of the heart, and then this layer here is all referred to as the epicardium. The epicardium again has multiple layers here. The outermost or superficialmost layer in this case is mesothelium, another type of simple squamous epithelium, but a type of epithelium that lines the outside surfaces of visceral organs and body cavities. So here you have the mesothelium. Deep to the mesothelium you have some connective tissue, and you can see some adipose tissue here. So if you see fat cells or adipose tissue, you know you're looking at the epicardium, not the endocardium. The endocardium is going to have the Purkinje fibers. In addition to adipose tissue and connective tissue, you can often see blood vessels through this region of the cardiac wall. You can also see some nerves, things like that. Here's another view of the epicardium where it's particularly thick. Lots of fat here. Here's a couple good-sized veins traveling in the epicardium. So make sure you can distinguish between epicardium and endocardium and observe the different layers of each of those tissues. Here is a high magnification view of the myocardium. You can see intercalated discs separating the cardiomyocytes, and you can see this is a good place to find lots of blood vessels. You can see lots of capillaries and small blood vessels traveling along between these cardiomyocytes. So that's it for the circulatory system. We're going to talk about the lymphoid system now. The lymphoid system is really arranged into in two ways. You have a lot of lymphoid cells, B and T lymphocytes, that are arranged throughout the body in tissues that are kind of embedded in other organs. This is sometimes referred to as the diffuse lymphoid system because of their diffuse organization throughout the body. The other part of the lymphoid system is organized into discrete lymphoid organs, in which you also have B cells and T cells and other cells. So we're going to look at both diffuse lymphoid structures and lymphoid organs. First, let's look at some solitary lymphoid nodules, which are part of the diffuse lymphoid system. These nodules are collections of B lymphocytes and nearby T lymphocytes that can be found in the walls of many organs, particularly in the gut, but you can also find them associated with the respiratory tract and other structures as well. So this is a solitary lymphoid nodule right here. This is in the gut of the colon. So here you can see glandular tissue of the colon. We're going to look at this in more detail next week. Underneath the glandular tissue in the region of the that's still part of the mucosa or the submucosa is a good place where you can find these lymphoid nodules. Here's a higher magnification view. They look pretty dark at low magnification because they are densely packed with B lymphocytes. B lymphocytes have, if you recall, very large nuclei that fill most of the cytoplasm and stain very basophilic. So these structures look quite basophilic when viewed at low magnification. There are both B lymphocytes and T lymphocytes associated with solitary lymphoid nodules. And B lymphocytes and T lymphocytes, as you may recall, are indistinguishable histologically using a simple stain like an H&E stain that's used for this slide. So the way that you can tell them apart is based on their distribution within the tissues, such that B lymphocytes are organized together in these nodular clusters. So this nodule here is composed of B lymphocytes. But if you look carefully in the spaces between the epithelial glandular tissue here, you can see dark nuclei of lymphocytes throughout this region as well. And these more diffusely arranged lymphocytes are actually T lymphocytes. Under a light microscope with this particular stain, they are indistinguishable. So you just really need to know them by location. B lymphocytes are always aggregated together frequently as these nodular type structures, whereas T lymphocytes are often more diffusely arranged. This is a section of the ileum where we have a macroscopic structure known as Peyer's patch. Peyer's patch is a collection of lymphoid nodules, so we would not call these solitary lymphoid nodules, but rather we have a cluster of lymphoid nodules. So this entire structure here is known as a Peyer's patch, and you can see it consists of multiple individual lymphoid nodules here. So there's about 1, 2, 3, 4, 5, 6, 7 or 8 of these, and probably more that continue off the section here that are embedded within the submucosa of the ileum. So take a look at this Peyer's patch structure in your particular slide. You'll need to use the slide that actually says Peyer's patch where that region of the ileum has been dissected out. There are other slides that show parts of the wall of the ileum that may not have Peyer's patch in it. So make sure you're looking at the correct slide. Here's a higher magnification view of individual lymphoid nodules within Peyer's patch. And in this case, you can tell that these nodules have a paler center that are lighter staining. This is an area of active proliferation of B lymphocytes that typically occurs when the tissue has been challenged with an antigen, perhaps through infection. And so when challenged with an antigen, these lymphoid nodules become activated and rapidly undergo proliferation in the center right here. So these pale staining centers of these lymphoid nodules are called germinal centers. And when a lymphoid nodule possesses a germinal center, we refer to it as a secondary lymphoid nodule. So make sure you know that term there. So these are all secondary lymphoid nodules that can be found in Peyer's patch within the ileum. Let me go back a couple slides. This solitary lymphoid nodule in the colon would be classified as a primary lymphoid nodule because it lacks a germinal center here. So keep a lookout for lymphoid nodules, solitary lymphoid nodules, as you look at various tissues in the remainder of the lab. You can find lots of them in the gut, but you can also find them in many other tissues, and so they're very characteristic by their basophilic appearance under the scope. Right now we're going to look at some specialized lymphoid organs. This structure here is called a lymph node, not a lymphoid nodule, but a lymph node. A lymph node is an organ that is surrounded by a connective tissue capsule, which is this eosinophilic structure that you can see right here. And all lymph nodes are encapsulated, and they have a couple of different regions. The outermost region, which is more basophilic and contains many lymphoid nodules, some of them secondary, as you can see here, some of them primary. This outer region that contains the lymphoid nodules is referred to as the cortex of a lymph node. If you look deep in the lymph node towards the middle region, you can see that there are no nodules, but rather cord-like arrangements of cells. This part of the lymph node is called the medulla. In between the medulla and the cortex, you have a sort of poorly defined region, which is known as the pericortex, which is in between those two. So be able to identify those different parts of the lymph node. Here's a higher magnification view of the outer cortex of a lymph node. Here you can see two nice secondary lymphoid nodules within the cortex that have the pale germinal center. These lymphoid nodules consist exclusively of B cells. T cells are found deeper within, either in the pericortex or in the medulla. You can also see the connective tissue capsule here. Note that the connective tissue capsule extends inward deep into the cortex of the lymph node at various intervals. These inwards extensions of the outer capsule are referred to as trabeculae. So here's a trabecula extending into the cortex. You'll notice that there is a space underneath the capsule and surrounding the trabeculae. The space is normally filled with lymphatic fluid that percolates through the lymph node after entering through afferent lymphatic vessels that you can't see on this section. The name of the space underneath the capsule is called the subcapsular sinus, and when that space extends around the trabeculae, it's called the paratrabecular sinus. You need to be familiar with those terms. Here's another section showing the cortex. Here you can see another secondary lymphoid nodule. Here's more of a primary lymphoid nodule. Here's the connective tissue capsule. Here's a trabecula, paratrabecular sinus, subcapsular sinus. Here's a higher magnification view of the medullary region or medulla of the lymph node, and so you can see that the arrangement of cells is quite different. Instead of these spherical or nodule arrangements of cells, you have these cord-like arrangements of cells. So these basophilic cords of cells are called medullary cords, and they're composed of both B lymphocytes and T lymphocytes, as well as plasma cells. In between the medullary cords, you have these fluid-filled spaces called medullary sinuses, which are filled with lymphatic fluid as it percolates through the lymph node. Here again is an even higher magnification view, and you can see cords of cells, medullary sinuses, as well as some blood vessels you can see here as well. So make sure that you can identify all the major parts of the lymph nodes. This is a section of spleen, and spleen is probably one of the hardest organs to understand histologically because it has a fairly complex structure. So I would certainly encourage everyone to read the chapter on lymphoid tissue in their basic histology text and pay special attention to spleen, because there's some really nice diagrams in that section that do a good job of explaining histology. So spleen, like lymph nodes is encapsulated, meaning it has a connective tissue capsule, a fairly thick capsule that you can see here, which again is eosinophilic due to the presence of a dense irregular connective tissue. Also, like lymph nodes, the connective tissue capsule extends inward into the outer portions of the spleen, forming structures again known as trabeculae, which you can see here. Here's a section of the inner part of the spleen, and spleen, the stroma or functional portion of the spleen is divided into two major parts. You have white pulp and red pulp. Let's talk about white pulp first. White pulp is basophilic when stained with H&E. It gets its name because it has a whitish appearance when viewed in a fresh specimen. That's due to the presence of white blood cells, which are mostly found in the white pulp. The red pulp gets its name because it contains mostly blood found in the sinusoidal capillaries in this region. And so it has a reddish appearance in a fresh specimen. With an H&E stained specimen, the red pulp is eosinophilic and the white pulp is basophilic, so it has kind of a deep purplish coloration. So this is all white pulp here and here. All this stuff here that's more eosinophilic is red pulp. Functionally, white pulp is organized into two ways. You have nodules of B cells. So lymphoid nodules are found all throughout the spleen and are a major component of the white pulp, and these are composed of B cells, not T cells. Some of the white pulp is actually composed of T cells, however, and these are found not in a nodular arrangement but as elongated sheaths surrounding arteries that are found within the spleen. So here you can see the wall of what's known as a central artery in the spleen, and you can see it's surrounded by a sheath of T cells, which we call the PALS, or periarterial lymphatic sheath. Sometimes the periarterial lymphatic sheath actually forms connections with nodular clusters of B cells. So if I am indicating this region of the white pulp here, this is B cells. This right here is the PALS, which is composed of T cells. Again, the cells are histologically indistinguishable based on coloration or morphology, but you can tell them apart depending on how they are arranged. Here's a higher magnification view of the central artery here. This region of cells that's forming a sheath along either side of it, this is the PALS, composed of T cells. But notice here is a nodular cluster of B cells which is sort of fused with the periarterial lymphatic sheath. This kind of looks like the wall of an artery, but in fact this is a trabecula here, and arteries pass through trabeculae initially, and trabeculae are not surrounded by a periarterial lymphatic sheath. So here's the lumen of a central artery that's surrounded by the trabeculae. So, but we don't call it a central artery in this case, we call it a trabecular artery. When it has left the trabecula and is no longer surrounded by that structure, it then becomes surrounded by white pulp. So here's another central artery surrounded by periarterial lymphatic sheath, and there are two nodules of B cells. So here's a lymphoid nodule consisting of B cells. Here's another one here. Here is a periarterial sheath consisting of T cells. Notice that in cross-section, the periarterial lymphatic sheath may look a lot like a lymphoid nodule, because now here you're seeing the artery in cross-section, and this is the sheath surrounding it. So it kind of looks like this structure, but because you can see the artery in the middle of it, you know this is a periarterial lymphatic sheath in cross-section. not a lymphoid nodule. Here's another example here. So for the structures we've discussed so far, make sure you can distinguish trabeculae and trabecular arteries from central arteries and periarterial lymphatic sheath and lymphoid nodules containing B cells. The red pulp is composed of two components. You have cords of cells, which are known as splenic cords, or simply cords of Billroth. In between those cords of cells, you have sinusoidal capillaries, which are very leaky capillaries. Sinusoidal capillaries contain red blood cells. This particular tissue section has been perfused, so many of the blood cells have been washed out. In your particular sections, you may see a denser organization because all of those cells may not have been washed out from the lumen. The cords of the cells, or cords of Billroth, contain a lot of different cell types, including B lymphocytes, T lymphocytes, macrophages, plasma cells, reticular cells and reticular fibers, granulocytes, a lot of different cell types in these cords. So they're fairly complex. You don't need to be able to distinguish specific cell types, but do be able to distinguish the splenic cords or cords of Billroth from the sinusoidal capillaries. So that's what makes up the red pulp. Next, we have a section of the thymus, another dedicated lymphoid organ. As you may recall from lecture, the thymus is a site of T cell maturation. T cells are produced initially in the bone marrow, but they then migrate to the thymus, where they continue to mature and be programmed. And so the thymus only contains T lymphocytes. There are no B lymphocytes, so there are no lymphoid nodules. You can easily recognize the thymus based on its lobulated structure, meaning that the stroma is divided into small cordoned off sections called lobules that you can see here, that are separated by connective tissue in between. And you can also identify it because of the dark staining, more basophilic cortex, and lighter staining medulla, which is deeper in the tissue. And the cortex, although basophilic, is not composed of nodules. There are no nodules to be seen here. Here's a higher magnification view of the medulla of a thymic nodule, and you can see this peculiar kind of whorl-shaped or circular-shaped structure that is found in the medullae of these thymic lobules. This is called a Hassel's corpuscle. Here's an even higher magnification view of it. And these odd structures, their function is not entirely known, but they seem to be involved perhaps with production of certain types of T cells within the thymus. But their function is not completely understood. And that's it for this week. See you next time.